In the first segment of this essay, I covered a more or less chronological story of my progress through learning about the science of DNA as it relates to Sasquatch. In the second, I talked more about the history of DNA studies that I've compiled and my understanding of them and what might work for moving forward in science. Here, I want you to have the best chance of understanding the rest by discussing the misinformed ideas that permeate the Sasquatch community. These are generally misinformed ideas that are actually common to people's understanding of DNA. It is merely exacerbated within the Sasquatch community the way they're used. I will repeat several important concepts throughout these installments of the essay. DNA is a really big topic, and the series is about as distilled as I could make it for what is relevant to this topic. Yet, there are many other important issues, so let me start by distinguishing between the terms disinformation and misinformation. I rarely use the word disinformation. The way I consistently use these words, this one involves the intent to disseminate false information, the intent to deceive. This is hard to prove, and so unless the evidence is overwhelming, I will never use or imply intent to mislead. There is no implication of intent when I refer to misinformation. An original presentation may be unintentionally wrong and misinform the audience. The presenter had no intent to deceive and was not aware of any incorrect information. Yet they were misinformed and, by definition, passed along misinformation. Anyone listening to that presentation will be misinformed and, if they share what they learned, will be spreading misinformation. It is only if you do fact-checking and discover the information was wrong and choose to pass it along anyway that you are disinforming those you pass it along to. It's only if you knowingly pass along misinformed facts with the intent to deceive that you are disinforming your audience. This is why I consider it vital for everyone to fact-check everything. It is a personal responsibility to ourselves and our community. It is important to communicate clearly what we believe and what we know to be factual in nature. We all know that what we believe is true. That's how wars get started. I understand and appreciate the passion that has evolved around this subject, but passion does not make one right. Science is a furnace in which irrelevancy is burned away to leave the precious facts that objectively define the properties of a discovery, while beliefs found to be wrong are left behind. Between the place we are now and the place we will be when we all understand the subjects better, we will all leave behind ideas we find to be wrong and adopt better ideas, as they are discovered, that describe more clearly the elements of these phenomena. The path to knowledge is littered with the shattered remains of cherished truths and the bodies of those who could not leave them behind. I now consider myself blessed to have missed the mess in 2013 because, as I began to more seriously pursue this subject around August of 2019, I was more of an archaeologist of the scene of the battle. I will refer to the Sasquatch frequently as the subjects, as in to say the subjects of my study in this area of knowledge. In part is to emphasize the idea that more than one thing may be going on. There are many phenomena that are superficially related to Sasquatch, and we do not know with certainty if all are truly related. Also, it removes a trigger word that will put up walls of thinking in the minds of more skeptical people or people who simply don't believe in the subject. So, referring to them as the subjects sets aside one possible trigger mechanism when you're speaking to people who don't know so much about Sasquatch. This is the most basic introduction I will do of the science of DNA. If you want to do more, All the information is out there, and I will help you find places to start if you really want to know more. The study of DNA, most broadly, breaks into two basic areas. Each of these is terribly complex if you want it to be, but complexity is in the eye of the beholder. First is the chemistry. Without the chemistry, you can't have any of the other information. The various compounds and procedures to isolate and separate DNA from other organic compounds for sequencing. Yet, unless you're on the boundaries of new technology, the chemistry is all well known and well established. All the necessary compounds can be specified for lab work to be done. Many can be easily obtained. Running certain experiments requires some specialized equipment for the work to come out properly, but from a certain point of view, the chemistry of DNA is no more complex than running a clean lab to prevent contamination of your work. Second is alignment. Once the lab has isolated, amplified, and sequenced strands of DNA, the fragments need to be compared to the databases of existing known DNA sequences. At this point, you're really just looking at arrangements of 4 letters, A, G, C, and T, mathematically creating a kind of a base 4 numbering system. Now, there are times when the sequencing is unclear, so there are special notations, different letters, that will represent uncertainty between combinations of A, G, C, and T in any point of the sequence. You will discover this if you dig deeply into the published sequences found by the Sasquatch Genome Project, SGP. Due to a variety of issues, there is some ambiguity in some bases, so a well-established notation, IUPAC, is employed. In the end, however, you're really just looking to match the sequence of these letters to determine how well they align, either with each other or with the databases of known genetic sequences. You don't have to do this yourself. There are computers doing this matching for you. One modern way to deal with DNA sequencing is to break all the DNA into fragments around 200 to 1,000 bases long and just sequence all of them as fragments. Then you use a computer to put the puzzle back together again by matching the ends of each fragment. If you match, for example, 30 bases from the end of one fragment to the beginning of another, the chances of that alignment being an accident are 1 in 4 to the 30th, about 1 in 1.5 times 10 to the 18th. These two fragments can be chained together in the computer to make a longer fragment. Rinse and repeat. This process is also used for environmental DNA, which I will touch upon later. This is computationally expensive, but chemically it's much simpler than the methods that were commonly in use up to 2013 when the SGP was doing its work. Once you have your reassembled DNA, you can compare it to the databases of known genetic sequences. When hundreds or thousands of bases match, it's almost certainly not an accident. Nature reuses DNA extensively across the phylogenetic tree. Thus, we can use these alignments to determine how a sample is related to the known branches of this tree. It is the oversimplification of this concept that leads to many of the misconceptions I'm about to address. So, let's get into them. DNA Misconceptions 100. Half human. This kind of terminology does not come from the study of DNA. It comes instead from the more traditional crossbreeding terms from the barnyard, like a half cow, half bison, or a half horse, half donkey, or a half goat, half chicken. From the standpoint of that sort of technology, chimpanzee cannot even be crossed with a human. Yet, from the standpoint of DNA, they are just under 99% similar. In this illustration, if the whole window is a human nuclear DNA molecule, about 3.3 billion bases, then the blue segments represent the amount of DNA that is different in a chimpanzee. The larger curve represents the relative size of chromosome 11, about 150 million bases. Chromosome 11 was used by the SGP in an attempt to amplify part of nuclear DNA from some of their samples. Looking at the blue segments here that represent the difference between humans and chimpanzee, If you were to look at the difference between human and Neanderthal or Denisovan, the differences are much smaller. They're far more like us than a chimpanzee. And remember, a chimpanzee is just under 99% similar to human. In fact, the creature we have DNA for that is least like humans is the octopus. Even that is about 85% similar to us. So, what does it mean to be half human? Anyone trying to use terminology like half human regarding an analysis of DNA is simply announcing that they have no idea what they're talking about. A related term, which is at least applicable, is a hybrid human. Yet, even that term should not be as shocking as you're probably thinking. Next time at the grocery store, look around at each person you pass by. The slight differences in the shape of the skull and the jaw, slight proportion differences in the length of fingers, the length of joints. Each of these variations is an indication of a hybridization event at some point in our past, with similar species. Many of these we only know because of fragments of DNA that we find in ourselves. Every one of us is, in fact, a hybrid human. So just get over it. DNA Misconceptions 101. The female DNA. Let's start with something that nearly everyone knows not nearly enough about. The differences between mitochondrial DNA, or mtDNA, and nuclear DNA, nDNA, in humans. We know that nominally mtDNA is conserved along female lines of inheritance. This is because sperm do not contain mitochondria. This has become twisted by misinformed people speaking as authorities on the subject of Sasquatch as the female DNA, which is balderdash. However, it gets worse because some extend this to say it is the female half of DNA, which is encroaching on just plain manure. In this illustration, once again, the whole white area is one human nuclear DNA genome, about 3.3 billion bases. mtDNA is a little over 16,000 bases. That's the little tiny black dot in the middle. Yes, it's actually there. If you have a large enough screen, you ought to be able to see it. It's basically only about 5 or 6 pixels. If the whole of the image represents the nDNA, then this tiny dot in the center represents a single ring of the mtDNA molecule. This little speck is what many so-called authorities are representing as the female half of DNA in humans. We'll get to the actual contribution of the female in the nDNA section below. Again, this is roughly 16,000 bases that do conserve along female lines. They allow us to trace migration patterns of human populations wherever remains allow recovery of mtDNA. mtDNA does not have so much protection from mutation as the nDNA, so it mutates somewhat faster. This allows researchers to identify haplogroups that develop within populations over time. This is specifically what allows tracking of migration with high reliability. The mtDNA is used for species identification of a sample because the molecule is relatively small for DNA and one cell contains up to hundreds of copies. In general, the identification of species can be established by recovering just a few fragments of this molecule called genes. At the time the SGP and the psych studies were conducted, this was mostly done with chemistry that's selected for specific genes. The chemistry varies between different species, and so these genes can be used to identify what species you're working with. When dealing with an unknown species, one tends to use agents that are more general in nature so that any genes that are approximately conserved between species will be amplified for sequencing. Note: one of the points that Dr. Ketchum ran up against was that all of the labs she used required filling in the species being tested of the sample. She frequently presented this as a barrier so labs could reject her kind of research. In reality, this information is used by the lab to select the optimal chemistry to amplify the genetic material for the researcher who's requesting the sequencing. When, in the first pass of the SGP experiment, the 111 samples all identified as containing human mtDNA, the first critical errors were committed. First was presuming that the DNA of the subjects was very close to human. While it fed into the narrative, confirmation bias, it was not necessarily a valid presumption. Second was specifying human as the species for those 18 samples selected for whole genome mtDNA sequencing. The selection of human-specific factors for the mtDNA sequencing of these 18 samples means that if there is any human contamination, it will be selected for preferentially. It also means that only genes conserved between humans and whatever else may be in the sample could be amplified for sequencing. As Dr. Hart explains in his book, this makes all the more challenging the task of identifying anything in the samples that may not be exactly human. Naturally, the results came back as essentially indistinguishable from modern human, which further fed into the confirmation bias of the study. DNA Misconceptions 102, Unknown Male. By this point, the die was cast for the SGP. Confirmation bias led to poor decisions in sample processing. Some samples had very little genetic material and were excluded from further study on that basis. Remember, the goal was to sequence nuclear DNA for Sasquatch, now believed to be essentially human because of cognitive biases from those driving the project. One cell has hundreds of copies of mtDNA, but only one copy of nDNA. The nuclear DNA molecule is almost 200,000 times larger than the mtDNA molecule. Obtaining an intact copy is far more complex and far more challenging than mtDNA. The nDNA is far more fragile, relatively speaking, in the wild when exposed to the elements. It is also far more sensitive to species-specific chemistry for amplification. The SGP selected chromosome 11 for examination. They did not state clearly the reasoning for this selection. The chromosome is roughly 5% of the nDNA molecule, if intact, around 150 million bases. There were 3 samples for which this extraction was considered successful by the project, 26, 31, and 140. However, the longest sequence successfully developed from this chromosome was only about 2.7 million bases long, just a tiny fragment of what would be expected for a truly successful experiment. The tiny little squiggle next to the very large curve is the longest base that Dr. Ketchum was able to amplify from chromosome 11, just a tiny fragment of the whole thing. So in this illustration, you can see that something was probably wrong. Using the chemistry specific to human DNA, the SGP failed to extract anything resembling more than a tiny fraction of chromosome 11. The sequence itself contains a number of possible clues for an entirely rational explanation of this. One of the most significant is the electron microscopy images in the study. They clearly show damaged strands of DNA, single strands where the pairs have split. These split strands will tend to recombine randomly in solution, as Dr. Hart explains in his book, meaning that any recovered sequence would be essentially random. Add to this the application of chemistry specific for humans, and you will only amplify fragments of the gene that are conserved between humans and whatever may have actually been in the sample. Here, however, is where the possibility of human contamination is likely to be overcome by the relative sample size. If the sample is large enough, then any shed human DNA containing it will be relatively small. As demonstrated by Dr. Hart in his book, it turns out these fragments align most strongly with a bear, a dog, and one of the researchers, respectively. I've confirmed those results multiple times using the National Institutes of Health nucleotide database. The fact that even the sample containing human DNA failed to amplify is a very strong indication that whatever nDNA may have been recovered in all of those samples was badly degraded. Some of the very evidence Dr. Ketchum provided to show that there was no contamination and no degradation, in fact, demonstrated the opposite, as pointed out in Dr. Hart's book. I also used the image enhancement software I have to make signs of the contamination in her evidence rather clear to see. It's easy to do. Look at her report, download the image, and start playing with gamma and contrast and color curves. So, where the SGP saw a mystery and almost magical result supporting their narrative, most people who examine the data from the context of trying to validate the experiment will see degraded DNA and possible human contamination in the samples, and will find supporting evidence provided in the published study. DNA Misconceptions 103, the male DNA. Well, we now extend the argument that the mtDNA is the female DNA, then nDNA is obviously the male DNA, right? Well, in fact, there is some portion of the Y chromosome that is actually conserved along male lines in this illustration. Again, to the same scale, the whole white area is the whole human nuclear genome, and the fragment in black represent the elements of the Y chromosome that are conserved along male lines. These can also be used to track human migrations over time. So, of the nDNA, this is the approximate portion that is exclusively from the male and is only passed to male descendants, as females have 2 of the X chromosome. Just an aside, as an engineer, while I was learning about all of this, there was one day I was having a rather amusing image cross my mind of God performing a post-manufacture check after creating Adam. I could imagine him finding that broken, stubby little Y chromosome and thinking, wow, how did that get past me? I'm going to have to fix that in the next model. Anyway, in truth, however, the female contributes about half the nuclear DNA, with the exception of those fragments of the Y chromosome. So, the actual contribution of the female nDNA in a male child looks a lot more like this. Note the implications here about the results of the SGP study. Because half the nuclear DNA is contributed by the female progenitor, a significant portion of any descendant would include her DNA, yet the SGP found nothing of the sort. They built their misinformed ideas that the mtDNA is the female half of DNA and nDNA is the male half of nDNA, which is misinformed at best and, frankly, deliberately misleading at worst. While the mtDNA follows the female line, because sperm do not carry mitochondria, nDNA is always a blend of both progenitors, except the portion of the Y chromosome conserved along male lines. Yet, the nDNA fragments published by the SGP show no significant signs of the presence of any contribution by human, male or female, even from the sample that is actually from a human who set the DNA trap in the first place. So in no way could anyone seriously claim unknown male progenitor when there is no sign whatever of the human female progenitor either. There is no mystery here. It is a failure to amplify, plain and simple. DNA Misconceptions 104, Sasquatch DNA Evidence. So now I'm going to return to one of my original statements in talking about the history of Sasquatch DNA studies that I've uncovered and investigated. There is no convincing DNA evidence for Sasquatch publicly available. If there is any privately available, it doesn't matter. Nobody learns anything from secrecy. Secret information is of no value to the community, and neither are those who keep it to themselves. The results of some DNA tests for species identification returning human is interesting. If enough material exists with these samples, if the material still exists at all, sequencing the whole mtDNA genome would be interesting to examine. It would most likely confirm human contamination of the samples, but if it was different, well, that would be very interesting indeed. Be aware that mitochondria are the engines that power our cells. If nDNA is the blueprint for a creature, then mtDNA is the blueprint for that creature's metabolism. In principle, the mtDNA of a Sasquatch could be very similar to that of a human, and that would indicate they are closely related not only phylogenetically, but would also be metabolically very similar to us. Still, it would be expected that their mtDNA would be more different from ours than Denisovan mtDNA is, but the SGP data doesn't support that, and no other study has bothered to sequence the whole mtDNA genome before the SGP, so they have nothing to present on this matter. DNA Misconceptions 105. The real next steps. Scientifically, the next steps are obvious if you're familiar with the science. With current technologies for DNA sequencing, whole genome mtDNA amplification is much simpler than it used to be, and is far more generic than it used to be for the SGP. The separate and distinct mtDNA of any species in the sample and their relative contribution to the sample can be measured far more easily. This would be more easily done than with nDNA due to the relatively abundant supply of mitochondria compared to the nucleus in each cell in any species. But now it's much simpler to audit everything in the sample and determine if there is anything more interesting in the soup. The problem of amplifying nuclear DNA is much simpler now. The systems will sequence even fragments of DNA and then work to align them with other fragments, creating longer strands and, with enough material, a whole nuclear genome to examine for everything that's in the soup. This will make it clear if they exist as flesh and blood, and how they are related to us and to all life on Earth for that matter. In the past, the greatest complexity was choosing the optimal chemistry to sequence DNA. Now it is the computing power that assembles these sequences from the fragments that we can find. For any lab, the identification of a new species would be a real plum. They would leap at the opportunity to participate in that discovery. So the question arises, if any samples actually do contain DNA from the subject, why has this not been found? Perhaps, just perhaps, no one has actually achieved a real clean sample of flesh from the subjects to date. Flesh would include blood, root on the hair, or actual tissue. Perhaps the handling of samples has, to this date, been insufficient to garner a viable sample to sequence. Perhaps, just perhaps, they're not actually entirely as we expect them to be. At this time, there's just no valid way to select the right answer. Environmental DNA 101. Sequencing a whole ecosystem. It's frequently asked, how soon should a DNA sample be collected to be viable? The correct answer is always, as soon as possible. Ideally, the subject comes into a lab to submit a sterile sample directly for verification. This doesn't always work, for example, with crime scenes. There, we have to collect the evidence at the crime scene. The guidelines are generally preferably within about 24 hours. We imagine that outside, in the wild, we would find samples contaminated much more readily, and you're entirely correct in understanding that. Bacteria will begin to digest the DNA within hours, and it decays quickly. Sunlight will break the strands of DNA. Oxygen will break the strands of DNA. Recent technology suggests that if a sample is collected within around 15,000 years, though, it may be possible to reconstruct reasonably long DNA sequences, complete enough to differentiate between species and even to identify new species. You actually heard that right, really. 15,000 years. Probably even longer, actually. Environmental DNA is a computationally expensive analysis process where a sample is processed to detect every fragment of DNA it contains. Any fragments long enough can be overlapped with other fragments in the sample, and they can be put together to create a longer sequence. Longer strands virtually in the computer during the analysis. We can be quite certain that these overlap because of the odds of an accidental alignment that I described earlier. Enough to identify mammoths, for example, even variations in mammoths, in eDNA samples taken in Alaska and Canada. Yet these samples were taken from Earth, from a mound immediately next to where the actual tissue is in permafrost. It's kind of like salting the mine, as it were. Yet the analysis of these samples, so far as anyone has said, have yielded no Sasquatch DNA in eDNA studies. This leads to a couple of conclusions that need to be investigated more thoroughly over time. First is, when these studies began to identify genetic material that strongly matches human, did they stop looking? Computationally, that's what I would do. If I know there are humans in the ecosystem and I start to identify human DNA, I don't need to match these sequences anymore, and I will set them aside. That seems perfectly logical and perfectly reasonable, and that might actually discard Sasquatch DNA. Or perhaps the Sasquatch DNA is simply not in these samples that have been processed so far. That's another possibility. As I mentioned, the mammoth DNA was collected from an earth mound. The mound was near the decaying tissue, so it's a lot more likely to contain a great deal of mammoth DNA and less of other DNA. Also, consider an eDNA study of Loch Ness. This yielded DNA identifying many species of fish known to be there, and of land animals in the ecosystem surrounding the lake, and no DNA from an unknown aquatic creature. It is worth noting that around 20% of the DNA was not specifically identified. These strands were too short to align properly with any others. They may represent unidentified microbes or bacteria or viri in the ecosystem. As I said, the process is computationally expensive. So I would not be at all surprised to find out that if a strand is identified as more than 99% likely to be human, they just set it aside and move forward. This is an idea that can be investigated by proper expertise at some point when they become interested. If you have some DNA that you believe to be from Sasquatch, if you have the sequence of the DNA, that is, I will be happy to examine it. So would Dr. Hart. I practice open science. Nobody learns anything from secrecy. At the same time, people have ownership of the intellectual property they produce. Even if they share it openly, there are legal rights that must be observed. I would produce a report based on the given evidence. I can keep identifying information secret. I have confidential information in a triple encrypted file that is never open when I am connected to the internet. I handle that information just as I would handle a root key for cryptography in computer data handling. Messages with identifying information are generally the only messages I delete, and only after preserving that data. I will produce a report. The report will be freely available. The report may be copied under the Creative Commons Attribution License identified in the document. Identification of areas and people may be abstracted as requested. The report will contain a report of the evidence chain and analysis of the handling of the evidence. I will consult other experts with general details for their opinions unless expressly prohibited. If identity information is part of the sample, again, it will be abstracted as a matter of process, but the details of handling and the details of process will be part of the report. The report will contain an analysis of the information offered from the samples. Again, I'll consult experts for additional analysis and interpretation as I deem necessary, unless explicitly prohibited. Since all the information will be available freely, other experts may also examine it independently. Principals for the purpose of such a report, and witnesses to whom the discovery of the sample is attributed, are involved in the collection of the sample. All those involved in the chain of custody, up to and including the laboratory performing the recovery of information, and those involved in the operation of the project, are part of the chain of evidence. I will automatically abstract all witness data. If witnesses provide permission to disclose contact information, a section of the report can include appropriate de-abstractions. If you require confidentiality, we can discuss fees for this work. My rates are very reasonable. Your project would be maintained in an independent, triple encrypted file for security. In the event of legal issues, this keeps your information independent from anyone else's information. Issues resulting from one project will not expose information from other projects. I still practice open science. I would prefer to share information that is relevant with the community, forwarding the understanding of the subjects, even if all the other information is abstracted for privacy. If all information of value is to be retained, my rates would be higher. I am not offering to pay for lab work to be done. I am talking about analysis of the sequence data already in your possession. If you have samples you want to be sequenced, that's beyond my current financial interest. However, if you want to preserve them for the future, or if you want to have them sequenced, we can discuss approaches for stabilizing the samples and preserving them and transporting them to a lab for possible analysis. I am the curious cryptid, and there is no disputing the existence of a set of phenomena we can wrap with the name Sasquatch. Unfortunately, the collective observations from several decades of all of these phenomena collectively still fail to yield significant information about the nature of the subjects. That alone is staggering. If the subjects are flesh and blood inhabitants of the real world, and I truly expect they are, we can expect they have DNA, and that DNA will place them in a specific location of the phylogenetic tree. I genuinely expect this outcome. If we continue to fail in recovery of DNA from the subjects, then we must ask why this is. It does remain possible that they are not exactly what we expect them to be.